just wanted to start with an acknowledgement of country. I respectfully acknowledge the traditional owners and the custodians of the lands and seas on which we meet today. I also respect that we have a number of parties joining from around the country today, and so would like to acknowledge the traditional owners and custodians of those regions and pay my respects to the elders past, present and emerging. For those who are in this room today, so Doug and I, we're meeting from the Miagin tribal lands and of the Turrbal and the Yagara people. I respectfully acknowledge their contributions. Thank you all for coming to this research showcase. Look forward to finding out what's exciting things have been happening within the CRC, and would like to now hand over to Cordelia.
Thanks, James. So welcome all to the Future Food System Research Showcase. I'm Cordelia Salomulya, the Research and Commercialization Director of the Future Food System CRC. Thank you for joining us today as we showcase some of the exciting research projects that are currently ongoing within the CRC. In today's session, you will hear from our 3 program leaders, several of our project leaders, as well as a few PhD candidates on topic ranging from urban planning for food and agribusiness, upcycling of agri-food waste, some of the latest development in indoor cropping technology that are specifically designed for the Australian soil and climate, and also state-of-the-art technology for food analytics for provenance and also added value products. As James mentioned, our CRC spans across the whole of the continent, from the East Coast to Western Australia and also Northern Territory. And we also welcome our partners that are here today and hope that this session will be very informative and useful for you. As a quick aside, last week I was in Chicago for one of the largest food industry event in North America. And the theme for that conference or for that event was how to future-proof our food system because of the concern regarding climate change that affect food production, supply chain issue due to geopolitical matters, decarbonization of the industry, and so on. So it is clear that these are challenges that are faced around the globe, and we are here trying to develop some of the answers through our CRC, particularly in the Australian context. Before we start, I just want to remind you again that this session is being recorded. Every speakers will have about 10 minutes for their talk, and there will be a Q&A session at the end of each session. If you have questions, please put it in the chat box and we will try to get it through the Q&A session. So I will now hand over the first session over to Professor Doug Baker, the lead for Program 1, for a short overview and also introduction of the speakers.
Thank you, Cordelia. Program 1, there's 3 programs. Program 1 deals with the larger spectrum in terms of the Future Food System CRC. So we look more at food clusters, food planning, food policy, urban food policy, logistics, and international trade. So it's a bit of a catch-all, but it has a large focus. So the speakers today, again, are quite diverse within Program 1. We've lee Lee-Jun Summerhayes, who's going to be talking about urban food planning in Australia. Professor Hun Han at UNSW will be speaking about smart agribusiness, the smart agribusiness sector in Namoi and his digitization project there. And lastly, Natalie Aaron is going to be providing an overview on her PhD of the circular economy framework in Western Australia. And I'll give more of an introduction when each of the speakers come forward. So first, Lee-Jun, you're first on, and she's just completed her PhD. It's under examination. And her PhD looked at urban food planning and urban food policy in Australia and whether we do or do not do it well. So Lee-Jun, I'll turn it over to you, and you could be the first speaker. And I think we're limited to 10 minutes. So take it away, Lee-Jun.
All right, I'm going to share my screen. Just let me know if you can see it. Is it showing up?
Yep. You got it.
Yeah. Excellent. Okay, yeah, thanks, Doug. Yeah, thanks for having me today. My name is Li Jun, as Doug just mentioned before. I'm a qualified and experienced urban planner and just finished my PhD at QT. And as part of the Program 1 for Future Food Systems, CRC, under the supervision of Professor Doug Baker, my research is focused on urban food planning in the realm of urban food systems. In today's research showcase, I will share some of my research findings and offer some insight on how does land use planning impact urban food consumption. So first, I'd like to contextualize food challenges on a global scale. We have challenges of rapid population growth and urbanization. Our planet is projected to have 9 billion people by 2050, with the world urban population continuing to rise from current 55% to 70% by then. Yet, urban population already consumes 70% of the food produced, and urban areas contributes to 75% of the greenhouse gas emissions globally. We also have ongoing challenge of climate change, both in Australia and other countries. We also face other mega trends, such as globalization, energy resource scarcity, and environmental degradation, as well as the COVID-19 pandemic and the war in Ukraine. So all these challenges inevitably placing increasing pressure on food provision, food production, and consumption. In particular, a recent report from the United Nations alerted a very slow or regressive progress in achieving the 17 Sustainable Development Goals set by 2030, while the global temperatures already hit 1.1 degrees. The report highlights several key areas lack of progress, such as for number 12, responsible consumption and production is attributable to insufficient policy development and implementation. And similarly, number 13, climate action is due to inadequate policy action. In addition, wealthy nations have been criticized for causing unequal food consumption and climate footprints around the globe. So this brings attention to food policies. Like many other developed countries, food policies in Australia are predominantly administered by the Agriculture Department at the federal and state level of the government. At the local level, though, the land use planning is primarily focused on regulating land use, housing development, or other aspects of urban development, but not on food. So to investigate the impacts of land use planning on urban food consumption, our research involved exploring the public perceptions of food concerning consumption by collecting 500 survey data collected in Brisbane between September and November 2021. So our research findings were achieved by analyzing 5 dimensions of food concerning consumption, and each one will be examining in detail. The first one is urban food security. Food availability was investigated by comparing 2 scenarios. As you can see on your left-hand side, 90% of people were confident with food availability in general. However, that confidence drops to 11% only when faced with extreme events, such as flooding or COVID-19 lockdown period. And this comparison raises a really critical question that how resilient our food provision in our urban environment. We also investigated the physical accessibility to food, and we found that 66% of people drove to do food shopping, and only 37% of people who are driving spent less than 5 minutes. And these results indicate the pervasiveness of car-dependent urban environments, and driving longer than 5 minutes also reflects the dispersed land use patterns and a lack of public transport networks. The economic accessibility is also problematic. 48% of people agreed with food affordability, in contrast with 90% of food availability as you've seen before. When it comes to the household spending on food, only 58% of people spent less than a third of their household income. The public's perception for food insecurity also returned 46% of people believing that this issue does exist in Australia. The second dimension focused on urban food epidemics by exploring the factors causing obesity or overweight. The first factor is food shopping preference. We only had 57% of people preferred to spend more on fresh and healthy food. The second factor, shopping frequency for non-essential food, returned more than 30% of people shopped quite frequently or all the time. So to understand the reasons for such shopping habits for discretionary food, the responses returned with 30% of people selecting convenient, 21% accessible, 20% available, 17% delicious, and 12% always on special, so affordable. It's encouraging to see that 82% of people aware of the consequences of over-consuming discretionary foods causing obesity or overweight. The third dimension is food justice, and we investigated culturally relevant and diverse foods by comparing its availability and affordability. Once again, descending from 72% to 42% represents a similar pattern to fresh and healthy food. And those patterns highlight that food affordability becomes an increasingly urgent issue for many urban consumers. We also explored various food rights and food charity experiences and found that 90% of people agreed that everyone has a right to access fresh and healthy food. 91% also agreed that fresh and healthy food should be made available, accessible, and affordable to everyone. Unfortunately, half of the respondents indicated food charity not available to them or the people they know when encountering financial hardship. The fourth dimension is about the food economy's negative externalities. We found that 78% of people shopped food in supermarkets. And this indicates the monopoly of supermarkets in the food retail sector. However, 63% of people expressed support for more supermarkets to increase competition, and 68% of people for more smaller scale food retail shops or markets. And those responses emphasize the issue of supermarkets' monopoly, as well as the subsequent reduction in food choices and diversity. The last one is food-induced ecological integrity. And public perceptions returned positive results across several aspects. The first one is that 71% agrees that the impacts of food waste and loss are detrimental to climate change. And 77% of people agrees that food companies should improve their business practices to address or mitigate greenhouse gas emissions. And 69% of people agree that food waste infrastructure should be provided in their neighborhoods. So by investigating the impacts of land use planning on food consumption, our research concluded several highlights. The first one is that land use planning without prioritizing food has negative impacts on urban food consumption. In particular, the dispersed land use patterns and low-density urban development can weaken food security and generate unsustainable food consumption. And finally, we suggest food-sensitive land use planning as a pathway to achieve the UN's goals of responsible consumption and production and climate action. So that's the end of my presentation. Thank you. And I'm going to stop sharing my screen now.
Thanks, Lee Jun. Are we going to take questions, Cordelia, at this point, or?
No, so at the moment, we are a little bit ahead of time, which is good because usually when we do talks, it's always over time. So thank you, Legion, for sticking to time. What we will trying to do is we will put the questions in the Q&A. So there's a Q&A button underneath, so you can submit your questions through that. And then we can have a Q&A session before the close of session 1.
Okay, well, if we're ahead of time, that's great. And I can see Hoon has joined us. That's great. So I'd like to introduce Professor Hoon Han at UNSW. And he looked at smart agribusiness solutions, digital agribusiness solutions in the region of Namoy in north central New South Wales. So Hoon, without further ado, I'll turn it over to you.
Oh, hello. Hi, everyone. Can you hear me well?
Yes.
Yeah. Okay. Yes. Thanks, Doc. My name is Hoon Han. Associate Director of City Features Research Center here at UNSW. This is an exciting project. Some of the areas I never discovered before, particularly agricultural business. But I trained as a planner and a geographer. This is something what I really enjoy that I like to proceed. The brief project background, you probably hear about the Namoy region. So it's vast area on central New South Wales around the Tamils. I guess you might be been in Tamils and around. There is huge investment in terms of agricultural productions, and they try to attract a new business. And often it's a difficult questions is those of newcomers and new business investors, where is the suitable land for particular agricultural business? And this project is developing supporting tool, decision-making tool for those planners and the new business or even tourists. They might interested in where is the suitable land for particular business purpose, and also types of agricultural product. So this is this huge fighting collecting data. So there is vast over GIS data and also location data, spatial data we need to collect. Apart from that, we need to understand those business types, agricultural business, what their scientific requirement. For example, beef farming, the water access and the slope is particularly important. But chicken shed is not. Its temperature is more important. So we need to understand vast, knowledges and in-depth knowledge about the science, but in the same time, we need to collect a large volume of geographic information. So that is what we are trying to do and develop the tool. So next slide, please. So we have a couple of major the step undertake in order to deliver the research output. First thing is that we collect the data, which include topological informations and business informations and all statutory planning and also land use planning. There's vast information we collect. Some of the data we couldn't collect it, which is we highlight later on. But one of the most important data we need, which is existing business location. So this is a huge land, huge area. We know that where is what types of business are located, what are the economic activities are. Once we understand these types of business, then we actually developed a lot of layers, geographic layers, and we actually analysis multi-criteria kind of analysis conducted, and therefore we can visualize the most suitable land for particular business, agricultural production, and so on. So this is the types of process we do. In order to do that, we need to know that what is the most significant factors affect land suitability. So we arrange and we actually held a co-design workshop with planners and industry partners and also experts in this field. So we have 2 workshops during the projects, and we actually conduct survey during the workshops, and those survey information, we actually developed a further refine and tuning our model later on. So in order to improve model reliability. So next slide, please. So this is types of business we collected. This is not easy because there is so many new business, existing business, and also the businesses haven't registered yet. So this is what we identified, about 6,438 agricultural related businesses. And you probably see that our largest number of business related to beef cattle farming is about 3,400 over. And there is different types of business. You can see pig, grains, and even mushrooms, a very small business, ships, and different types of business. Each business location we actually geocoded. So we actually create a map which we can access information where the business are. So next slide, please. So this is the location of different types of business. That maps is also available using the website. Your map link is there. There is Kepler we use. This is kind of customer version. So you wouldn't really see the very detailed information, but it's enough to see that basic information, sense layer, and the location, and so on. So this is hopefully might be able to zoom in, but unfortunately this slide is fixed screen. So if you click each dot is show the information, what types of business, and business names and location and so on. This little box in the middle, you can see that different colors means different business types and so on. So next slide, please. So we create a bit of a density map. This is kind of a good exercise. Once you understand whether it's certain types of business they like to cluster. For example, cattle farming, they tend to clusters. If you find one cattle farm, your neighbor might be do similar business. That sort of things. It's about chicken shed, it's just random. You can find it anywhere. So this is a sort of understanding what different business, they might have some sort of pull and push other business. So we try to allocate this business and generate the density map. So this is a little bar, is to show that what types of business is sort of number of business, and the low business, different lengths of bar, kind of 3D bar chart, which is to show that the business density and concentration and so on. Next slide, please. So it's another way we can show that cattle, beef cattle farms. This is you probably see that about 3 major kind of clusters around Tamers, and those the area is actually existing cattle farm. So why we actually need to understand existing cattle farm. If someone actually want to have a new location of a cattle farm, this is actually relatively a good reference. This is types of good suitable land for cattle farm because they already have the infrastructure and service available around there. Then this is also kind of show that there's less risk natural hazard, for example, flooding and bushfire and so on. So this is our types of business. Each business, we try to understand their location patterns. Then we develop our decision-making tool later on. Next slide, please. So this is a types of layer we developed in a multi-criterion layers. I might show that different layer what we use. and how we actually use this layer to develop the multi-criteria toolkit. So next slide, please. And this is types of the criteria what we need from literature review. For example, is crops, is soil quality is very important. You know, your HP level and hay pasture. You know, land. This is a soil moisture is important for climate, and for soil texture is quite important. So a lot of science is behind that. So you try to understand each business what is significant, you know, criteria for the business. For example, the slope is very important for beef and lamb and pig meat. So we collected most of this data through the very different sources, range of sources, you know, government website, open sources, and also some of the data we actually purchase and order it. But one of the data we couldn't access, which is groundwater availability, which is unable to access, and other important data like odor, because it's the neighborhood, the odor that is in particular, you know, cattle and those pigs and chicken farm area, that is odor is particularly important. We couldn't access this odor emission, unfortunately, and there is some limitation I want to say. Next slide, please. So this MCAST tool, which is a kind of software, we actually need to develop the algorithms and the formula behind. So this is a kind of, you know, the a platform we use, which is an open source software, but we need to develop, you know, all the data layers. So we use about more than 30, 40 different data layers. And also we need to provide, this is a weight, each weight of each criteria. So, and then, you know, actually we use this multi-criteria analysis, which turn into visualized most suitable land for particular business. Next slide, please. Okay, this is the weighting method. So it's basically, you know, apple to orange. It's very similar. What is soil is more important than slope. So this is a types of analysis. First, we screen through the literature review, and we actually make a criteria, which one is the most important variable. Then we go through that co-working design, those experts, we conduct a survey, and basically it's a pair-wise survey. Okay, soil types and the soil slope, which one is more important? This kind of give a scale, and each expert actually give a slightly different scale, or if think there's more are important, is probably in the middle. So we collect this information and update this weighting later on. So this is called AHP method, but I wouldn't want to go through in detail, but this is a simple explanation. Next slide, please. So this is a co-working workshops, and James and Doc was there, and Maria was there, myself. So we invite, you know, a couple of city council planners and business partners, expert there. Then we evaluate the criteria, which criteria is missing, what criteria is more important, more weight than the others, and so on. So in the during the workshop, we found a very interesting the view. So next slide, please. So if you see, this is a types of a word cloud, and that we ask, you know, what factor is most important in particular business. Poultry, you probably see that the water and transport is sort of important. And you know, cropping, climate, and soil is very important. So different word is more than 230 different, you know, the criteria. The expert is actually type in online survey. So they is automatically generate these types of word cloud. And we found that some of his livestock is transport is very important, which I didn't know. And so we try to looking at those accessibility to major kind of transport hub and service center and so on. So this is a very helpful practice. And next slide. Then we actually weight each criteria, which one is higher and the other. So this is a kind of weighting matrix. So you probably see that the higher number is extremely important than the others, and the less important. So this is a metric actually used, develop some sort of formula when we actually developed this tool. So this tool actually use these types of metrics to generate most suitable land. When they evaluate different criteria, they actually suggest different types of land use and land suitability for particular business. Therefore, each business type has a slightly different land suitability map. So that's what we actually visualize at the end. Next slide, please. So this is a types of practice. We show that transport access where is more easily accessed or very hard to access in high slopes is still no arterial road or truck can't access particular region, and small 2 lanes and 4 lanes in 1 lane, and etc. And utility accesses is in particular service centers and transport hub, and major utilities and waters and so on. And the natural environment, obviously bushfire is big issue, and a bit of flooding area as well. And local, kind of, context, land use planning, the easement and buffers, and some environmental protection, conservative land, and so on. So this is we use all different types of layers and generate the map. Next slide, please. You see that this is a types of tree, and this is the software automatically detect what sort of the cluster of criteria. For example, soil types and soil humidity is a similar cluster. They actually cluster the soil types of evaluation criteria. Then climate changes, then we use temperature, humidity. This is a climate change cluster. Then they automatically decide which cluster combine all together. The map is developed, medium map, and the final map we consider everything is together. So this is a kind of a different stage developed how to actually develop the land suitability. At the end, we consider all in sink, kitchen sink types of analysis. If we consider all variable, different weight, okay, and what is land suitability looks like. And next slide, please.
Boone, you've got 1 minute left.
Yeah, so almost done. So this is kind of map suitability of livestock farming. This is the area is more suitable and not suitable. You obviously see those red areas not suitable, and the suitability map, which is available again, the website, the internet link, the map link, and left-hand side. Next slide, please. And this is our final slide. So we generate each land suitability of particular map, which is useful for business investors and planners, help them make decisions of future land rezoning and business cluster by they looking at. So this project is finished about just 2 weeks ago, and the final report is a handover. So we testing these various scenario at the moment. Then we probably need further tuning once we get the feedback. So this is our project quick summary. Very happy to answer any question. Thank you.
Thank you, Hoon. The rule is the more data, the longer it takes to explain it. So thanks, Hoon. Our next speaker, PhD student from University of New South Wales, Natalie Aaron. She's exploring the carbonization of magnesium-rich brines into high-purity hydrated magnesium carbonate materials, which can be used as sustainable building materials. She's working with one of our partners, EcoMag, out in Western Australia. So Natalie, I'll let you take it away.
Okay. Can everyone see okay? Okay. So I'm Natalie, and yeah, my project is on the circular economy framework to manufacture sustainable construction materials from agri-food wastes. Just for some background, the linear model used by the construction industry is obviously not very sustainable, and as a whole, the industry accounts for around a third of natural resource extraction and a quarter of solid waste generated worldwide. So a more sustainable model would be the circular economy framework, which decreases the volume of waste produced and lowers the amount of resources that have to be extracted. And EcoMag, which is our industry partner for the project, uses a manufacturing process for their hydrated magnesium carbonates that does fall in line with the circular economy model. So some more about EcoMag. They are a CRC industry partner, and they're looking to broaden the application of their hydrated magnesium carbonate and magnesium dihydroxide products. And their method for making hydrated magnesium carbonates looks something like this, where you have a desalination bittern that's rich in magnesium, you add sodium carbonate, and the product that's produced is niskeenite, which is a form of HMC. This process can be used to make materials that would be able to penetrate the construction market given the drive for more sustainable options. And it has the potential to integrate carbon capture as well as agri-waste materials. So the aims of the project are to integrate sustainable practices into the manufacture of EcoMag's construction material product, progress the manufacture of the material from a laboratory to a prototype scale, and examine the impact of including additional waste streams like producer gas and agri-waste into the final HMC product. So overall, we're looking at designing a system kind of like this, where you take captured carbon dioxide from producer gas with sodium hydroxide to make the sodium carbonate, and then applying it to EcoMag's process with the bitterns to produce niskeenite. And then there's some post-treatment processes that I'll talk about later that lead to the final HMC-based product that can be used as a substitute for plasterboard. So step one, the carbon capture. This is a diagram of the CO2 sparger that's been fitted to a reactor at EcoMag's pilot Chatswood site. Right now it uses pure CO2 as a source, but the goal is to eventually use something like a producer gas, which is made from biomass gasification, and it'll have other contaminants like carbon monoxide, methane, hydrogen, along with CO2, but the effect of using a gas mixture instead of pure CO2 hasn't yet been investigated. Using sodium hydroxide instead of commercial sodium carbonate is slightly more expensive, about 10 cents more per kilogram, but the market research from EcoMag suggests that the cost can easily be absorbed. So the next step is the synthesis of niskeenite. Niskeenite is one of 3 phases of magnesium carbonate that we're interested for this product. Niskeenite is the precursor, it's easy to synthesize. Dipengite is a transition state. It's not as easy to synthesize, but it is slightly more thermodynamically stable than niskeenite. If you just leave niskeenite out, it will slowly transform into dipengite, and then from dipengite into the final phase, which is hydromagnesite. It's the most thermodynamically stable, and it's likely what the final material would be mostly composed of. So niskeenite, like I said, it's synthesized really easily, and using EcoMag's method, we can achieve an extremely high purity. It does need to be dried slowly in the first step in order to avoid a premature transition, because the drying and treatment conditions of niskeenite are detrimental to building the compressive strength in the final product that would make it actually usable. So the next step is those post-treatment conditions, the thermal activation and curing. So thermal activation is based on the fact that niskeenite can be reconstituted after partial dehydration. And using a dehydration and rehydration cycle can build compressive strength in the product. The highest reported is 15.6 megapascals, which is well above what a typical gypsum plasterboard would be, which is normally in the range of 2 to 8 megapascals. So looking at the thermal activation here, it's the temperature after drying that's the most important, because the temperature, the thermal activation temperature, and the temperature released after rehydration is a positive correlation to the compressive strength. So if you have the thermal activation temperature too high, then the water will be driven off too quickly and the strength won't develop. You'll just keep a loose powder. So really understanding the relationship between the thermal activation and the rehydration reactions is going to be key to building a strong product. And the exact conditions needed for the product are we're still investigating. So what happens during that partial decomposition? Like I said, it's going to be key to understanding that mechanism is key to understanding how and why the properties develop. So the minimum that we found is 12 hours at 75 degrees C, which drives off approximately 1 mole of water. And if you look at the X-ray diffraction data, we can see that the crystal structure has started to decompose. To get some more details on what was happening, we looked at the FTIR, which showed a peak appearing after 12 hours at around 972 centimeters -1, which is consistent with a bicarbonate group rather than a carbonate group. So niskianite has the magnesium carbonate structure, and we've theorized that it decomposes to a bicarbonate structure with 1 less water. But again, FTIR also has limited details, so there's still more investigations to do into that to confirm if that is in fact what's happening. So how does agri-waste tie into this project? Crop waste is often discarded or burned in an unsustainable way, but agri-waste ash can be CO2 reactive. The CO2 uptake potential varies widely depending on the material, anywhere from 4 to 30%. These ashes have been used in construction materials before. Rice husk ash has been used in Portland cement with no negative impacts up to 10% substitution for standard pozzolan. And you can use it up to 25% with limited impact on the compressive strength. There's a slight decrease, but it's not too significant. Another positive of using biomass ash in this material specifically is absorbing the CO2 that's released during the curing process. When niskianite transforms into dipengite and hydromagnesite, 1 mole of CO2 is released for every mole of hydromagnesite formed. By integrating CO2 reactive ashes into the product, we could potentially absorb that CO2 before it's released, which would further lower the carbon footprint of the final product. So the carbon footprint, this is the life cycle analysis from Ecomag on their HMC production process. So what this can tell us is a couple of things. First, the net carbon footprint is largely dependent on the source of sodium hydroxide. But this system does have potential to have a lower carbon footprint than a standard gypsum plasterboard. However, this model has not included the addition of agri-waste, largely because we haven't decided on a source of agri-waste yet. And because of that wide range of carbon uptake potentials, it's hard to calculate something yet. But overall, it does show that there is a large potential for a low carbon construction material. So what's next for this project? Like I said, further investigation into the thermal decomposition mechanism in order to find the ideal thermal activation and curing temperatures. Further testing on the physical properties. Right now in the literature, there's really only data for compressive strength of these materials, but they do have potential as fire-resistant materials, and there's more information needed about the thermal insulation capabilities. And 3, to examine the impact of integrating more waste streams like agri-waste ashes and producer gas mixtures. That's all I have. Thank you.
Thanks, Natalie. Well, that's right on time. Those 3 talks really show you the diversity of this program. It's wide-ranging. So I have a couple of questions here. The first two are to Li Jun. Think of some questions, you guys. We have time. The first one is interested in hearing your thoughts about how we might go about implementing better urban food planning in Australian cities. Li Jun, any suggestions?
Yeah, there's many suggestions. However, I want to emphasize that agri-food systems are dynamic and multi-dimensional, and there's no one solution can one size fits all. So this is where my research is focusing on the urban food system to examine this issue, how we can go about, you know, from the perspective of policymakers and professionals working in the urban environment to do something to improve the food system in the urban perspective, especially how do we make it more sustainable, resilient, healthy, and equitable for everyone. In terms of the suggestions, part of my research shows that the policy in terms of from the planning perspective is not adequate enough to address food and understand the importance of the food systems in particular. As mentioned in the slides before, that food is predominantly addressed by the agricultural sector in the federal and state level of the government. From the land use and planning perspective, there's hardly any policy specifically addressing it. So we have a long way to go to really become, you know, to improve our food system from a better way. One thing I would suggest that, yes, we need to look at all these multi aspects, including how do we use better use water, land, and how do we make the housing, you know, more densified in certain areas? How do we improve the transportation networks in the urban areas as well? Yeah, I hope that's I think it's multifaceted, and I think to think about the short time period, it's too hard to answer the question. Yeah,
that's a tough question, Li Jun. There's a couple more for you. The first question is, how should land be used, particularly when it is scarce, i.e. repurposing highly fertile agricultural land for housing? But a second question ties to this. Did your research look at the importance of peri-urban areas for food production, which are often victims of land use conflict and urban encroachment? So it's the same kind of juxtaposition.
Yeah, it's really tough questions, and it's a challenge being faced by many planning professionals as well as the policymakers, trying to balance between build more houses to accommodate the population growth, or do we need to cultivate more land to support the agriculture economy and relevant food production? It is a tough call, and it's ongoing challenges and involving multiple stakeholders to work together to address this issue. Yes, we also think about climate and other energy. I know, you know, we only have 2.5% of water, fresh water, drinkable on the whole planet. And how do we, you know, use water properly, as well as the scarcity of the land? It is a tough call, but a few suggestions from my research shows that, you know, we could improve the more mixed use the land use in certain areas, so we can optimize and mix with different retail, housing, as well commercial and industrial uses together as one goes to optimize the use. So I hope that I have answered the question.
There's one more, and it's best practices. Can you recommend some best practices that you've researched? Where would we look for best practices? You've got about, I'll give you 30 seconds.
The best I would recommend is the integrated policy development and implementation, as well as the governance approach. As I said, it's not in the urban areas.
No, where are best practices? Where can we look to best practices? Geography. Where in the world? There's
some case studies I've been looking at from Canada, I have to say, yeah, like Vancouver, and even some practices even in New York as well. So there are something happening, and I have to say, compared to the other countries, Australia is lagging behind.
Okay, good. Any other questions for the other speakers? Anything for Hoon Han? Ask him a tough one. Or for Natalie. Anything? All that hard work, Hoon, and they're not going to ask you anything?
I have a question for Natalie, I guess.
Good.
How much CO2 can be absorbed by putting waste into the materials that you guys are producing?
So the final product, hydromagnesite, has a ratio of magnesium to CO2 of 5:4. But the total amount of CO2 absorbed is going to largely depend on the additives. So how much additional CO2 can we absorb using something like biomass ashes?
Right.
And that isn't something that we've really started to get into the details on yet. But it is something that we're planning for.
Yeah, it'd be a good calculation to make in terms of sustainability and, any opportunity we have to absorb CO2 or reduce its loss to the atmosphere would be good.
I think we're almost out of time. It's 2:53. We're 2 minutes ahead of the schedule. Shall we turn it over to program 2, Cordelia? Are you okay with that?
Yes, there's actually one additional question for Hoon, just come last minute. So
Oh, yes, there is. Yes. Shall I read it? What will be the implication of your research on merging or expansions of agri-industries in Namoi? The RJP is a job precinct with a view to grow economic growth and jobs.
Yeah, I think these are excellent questions. In fact, the Namoi region is, I found that it's actually the job accessibility is another region, those peri-urban area, as you know, that is population lost, and also there is the economic activities is declining over time because of this metropolitan types of the urbanization in Australia. So government policies really focus on encourage those regional economic activities and also economic growth and business activities and so on. So what actually I found that here is that there is a huge opportunity in the agricultural business. The people often don't know the region area in terms of its planning system and policies and types of economic productivity and so on. There is a bit, I think it's a bit hesitations there to move to these regions. But one of the things is I think I found that given this kind of supply chain issue in Australia, and its agricultural business is have a lot of potential. You see, we found that the food prices goes up, and how we actually provide a sustainable food for the Australian. And this is I think the area is we need to look at. This peri-urban area is a regional area in Australia, and we need to invest in terms of economic activities and business opportunity. Therefore, it's create more job opportunity in there. Okay, so that's a kind of implication I would like to extend. How this is types of the studies help encourage the more job opportunities and attract more people in regional area.
Good. Thanks for your question, Brooke. Thank you, Hun. All right. Well, I guess I'll turn it over to program 2 to distinguished Professor David Tissue. DT, it's
Thank you
yours.
very much. I'll give a brief overview if Luke could load up the slides for me.
Always got slides.
Yeah, I had to do one better than you, Doug. That's my only requirement here. Program 2 is largely about protected cropping. In terms of the program overview, a few of the things that we attempt to do in this program is to make protected cropping more productive, cost-effective, and sustainable. We try and increase the amount and quality of the crop for domestic and international markets. Develop technological solutions primarily around sustainability, that is increasing energy, water, and nutrient use, and thereby reducing operating costs. To develop or improve modular growth systems for use in high-density urban areas like vertical farms, and reduce the distance to markets by placing our structures in peri-urban areas. And lastly, to sort of take an approach where we utilize robotic and sensor systems like Uliganoom's new project to maximize the detection of pests, crop quality, and harvest crops while minimizing personnel costs. Next, please. These are examples of the current projects that we have in Program 2, and it's fairly substantial. I think I've gotten most of them. The ones in red are the ones that we're going to hear about today. I selected numerous microbial rhizosphere projects because we've heard a lot about plant production, but we've heard much less about what's been happening below ground and how that can improve crop production and also pests and so on. So these 4 projects are ones that we're going to hear about in that order. Next, please. That should be the end, so we can get rid of that screen. Thank you. So what we'll do is we'll end up having 4 talks. The first talk will be from a student, Phil Thomas at UNE, and he'll talk about microbial rhizosphere diversity in glasshouse hydroponic crops. Phil?
Thanks, David. G'day, everyone. Am I coming through okay?
Loud and clear, Phil.
Yes.
Just switching to presentation mode. Okay, so my name's Phil Thomas. I'm a PhD student at University of New England in Armidale. And I'm working on this project, which is looking at the microorganisms that live around the roots of hydroponic tomato plants. So we're switching from the broad scale to the microscopic. So hold on to your hats, do not adjust your sets. So this is a project which is part of the future farming program of the Future Food System CRC. It's a collaboration between Costa Tomato Exchange at Guyra and UNE and Western Sydney University. And our ultimate aim in this project is to develop a test for the microbiome in the hydroponic systems that the growers can use to track the health of the microbiome and hopefully get an early warning of any problems appearing in the crop. So we started the project back in 2020. One of the first things we did was to set up an experimental crop in the UNE glasshouse. And we aimed to make an environment which was as much like a Costa commercial crop as we could. So using commercial varieties of large truss plants and grown to maturity and fruiting before we started our study, and growing in the same kind of substrate, using the same kind of irrigation and maintaining the crop, picking and leafing and etc. Because all of these factors, the environment, the plant, the maturity of the plant, have a big bearing on the makeup of the microbiome. So we also study crops in the Costa glasshouses. So in 2021, we sampled a commercial crop again of the large truss tomatoes, which is the most economically important for tomato growers. And we sampled it for the whole crop lifetime, starting in January with the baby plants and finishing after the ethylene addition and just before the final harvest in November. So 16 time points, 500 something samples. And this crop turned out to be a particularly successful, healthy crop. So the data that we've extracted from that now is our reference for what the microbiome of a healthy large truss tomato looks like. So we also, this activity is just finishing up in the UNE glasshouse right now. So in contrast to the healthy crop, we're looking this time at the microbiome response to tomato plants that have a pathogen problem. So again, this is the same kind of environment, large truss plants and grown to maturity before we started treating them. Started last year, and around the end of last year, I started trying to infect powdery mildew into the crop. It turned out to be a little bit different, a little bit difficult. And just as I was giving up after the second inoculation, we managed to get powdery mildew into that crop. And we also then took some Fusarium pathogen spores from one of the Costa glasshouses and managed to get a infection symptoms coming up in the crop as well. So the samples of this experiment over this time are still in being processed. They haven't been, we haven't done the DNA sequencing yet, but this will be an important contrast to our healthy crop. So what have we found about the hydroponic microbiome? Well, one thing is that it's highly consistent throughout the glasshouse, which is, as you would expect, in a highly controlled, protected environment, and it changes gradually during the lifetime of the crop. That's also what we expect because the plant development and changes in the environment affect the microbiome. But the fact that it's a consistent and gradual change is good for our test, because the more consistent the healthy signal, the easier it is for us to detect deviations from that. The other thing we found that was expected is that there are distinct communities living in different locations in the slab, and that's as a result of different environmental conditions, pH gradients, EC gradients. And the fact that we can clearly distinguish those is also good for us because it shows that we can detect changes in the microbiome coming from the environment. Now, when comparing to soil, we found something that we didn't expect, and that is that there were more species in the hydroponic system in the root zone than we found in tomatoes growing in soil in a high tunnel system. So similar kind of commercial large truss tomatoes. But there's a different balance between the two systems. And there's also a very big difference in the type of organisms which are dominant in the two systems, which you would expect. So the hydroponic system is not what you may think a clean, not very many organisms, not much variety. It's actually as complex as a soil rhizosphere, but with a completely different environment, a completely different makeup, and very different organisms. So possibly worth investigating. Now, the approach for our health test is based on the principle that the microbiome is closely attuned to the plant, especially in the hydroponic system where all of the nutrients for the microorganisms are coming from the same kind of plant, the same crop. And if the plant is affected by stress, nutrient problems, pest, pathogen attack, it can change its root chemistry, and that affects the makeup of the microbiome. So if we can detect reliably the signals that indicate the deviation from a healthy microbiome, we can give the early warning to growers, gives them the opportunity to do some increased surveillance, early intervention, and a week or two early warning of problems in the crop is a very big advantage in the hydroponic system, where the difference between snuffing out a problem before it spreads, before it gets out of control in the glasshouse, makes a huge difference to the overall yield of the crop. So that is, in a nutshell, the hydroponic root microbiome project. So thanks everyone for joining the presentation. Thanks everyone who's been involved in this project, and happy to take any questions.
Okay, thank you very much. If you could stop sharing your screen.
Yep, just as soon as I find where it is. There we go.
Beautiful,
Okay.
nice work. That's great. Thank you very much, Phil. That was quite an excellent talk. I think what we'll do is we will save all of our questions for the end. That way, we'll make sure that we get through all of our talks, if that's okay.
Yep, sure.
Okay, thanks. Okay, so our next speaker will be Professor Zhenghua Chen. He's in the School of Science at Western Sydney University. He's going to talk about sustainable fertigation for high yield and quality vegetables in protected cropping. Zhenghua?
Thanks, David. Okay, I'll share my screen. Okay. Share.
Please remember to put your questions into the chat Q&A. Thanks.
Okay, can you see the screen now?
Yep.
Okay, that's good. Good afternoon, everyone. As David just mentioned, I'm from Western Sydney University. So this project is a collaboration between Western Sydney University and Qatar University in Qatar. And we're looking into some good strategies to reduce fertilizer use. Okay, so as everybody knows that fertilizer are really vital for agriculture, but we have those current issues, and I think this is not the first time. Here, just a few graphs showing the fertilizer supplies, and Australia is a net importer of fertilizers because we only produce a fraction of fertilizer for our agriculture, but our agriculture, including protect cropping, has been heavily relied on fertilizer imported from Russia, from China, from other parts of the world. And the COVID pandemic and the recent Russia-Ukraine war had made it more difficult, because you can see here the prices as shooting up at the historical high for all fertilizers. This really reminds us that in order to achieve high yield and quality, and we also need to look at whether the growers can actually make a profit. You cannot use as much fertilizer as you want or as you used to. We need to find ways to reduce fertilizer use. And this is just a table showing which countries are the producers, and you can clearly see China, India, United States are the top three producers of users of fertilizer, and they also, especially China, export a lot. Then Russia is also another country combined also export a lot. But Australia is, as I mentioned, is a net importer of fertilizer. Okay, so in Australia, protected cropping, the industry has been very successful and has been continuing growing at a rate of probably 5% per year. And we see more and more greenhouses and high tech and mid tech and low tech being put up. And primarily, the focus of the production is really looking at the yield, and the quality has been now been really looked by researchers and also growers to improve the quality, but yield is the primary goal. And to improve those yield and fertilizer, or in protected cropping, we have another term, fertigation, fertilizer plus irrigation, is one of the key inputs. The fertilizer and the transport fertilizer from one country to another country, especially to Australia, this is an issue. And we also have another issue is the Australian dollar has been the value has gone down against US dollar and other currencies. So this means growers have to spend more money on fertilizer if they continue to use the same amount of fertilizer. And in Australia, there are no breeding programs, so we cannot really breed varieties with more fertilizer use efficiency. I think in other countries, like in the US, India, and China, they're doing this. But of course, we can import those varieties. But one of the good way for reduce fertilizer use, or for keep the high quality produce and also maintain the yield is through management, especially the sustainable management of fertilizers. So just go to the aim of the project. This project, we try to identify some novel fertigation, nutrient delivery, and management approach to grow vegetable crops, mainly capsicum, eggplants, and cucumber. And we also have a specific research question look at whether the reduction of overall fertilizer use and such as potassium and nitrogen can still maintain the yield and quality, taste, and nutritional value in greenhouse crops. We've conducted a few experiments already. So we measured photosynthesis, yield, mineral nutrients, fruit quality, and gene expression in terms of qPCR, but we're also planning to do transcriptome analysis and metabolites to look at the actual quality attributes in those vegetables. And then another important part of this project is using panels of potential customers. So we probably use students to do the sensory evaluation to look at the quality. So far, we've done 3 experiments, 3 trials, 1 eggplant trial, 1 cucumber trial, and 1 capsicum trial. And we're planning for a few more trials. So those in the dark are completed. We have different electroconductivity levels, depend on the crop. We will run a potassium and nitrogen trial in the next few years. So just quickly go through the results. Eggplants fertigation. This is an early trial during COVID, but we still managed to go through the crop. But the data collection was a bit limited, and we still haven't really finished all the analysis. But we're planning to analyze and publish it hopefully by the end of this year. So effectively, what we found is high and low fertigation do not really affect growth yield of eggplants so far, and the two variety, Lydia and Tracy. If you look at the growth rate, fruit number, and even the gas exchange rate, the difference is not that great. Even for fruit weight, for some reasons, a very high EC actually reduced the fruit weight in one of the varieties, Lydia, but not really affecting Tracy. And we also did some correlation analysis. As I said, the results are still being processed. The second trial, the capsicum fertigation, was conducted in 2022. 2 varieties. The red one is called I can't remember, Tracy, one of the name. And the yellow one is a variety which has not been released by Syngenta yet. So what we found is a high level of fertigation does not improve yield and sugar content in both red and yellow capsicum varieties. In some cases, even the high EC value actually reduce yield. So that means higher fertilizer is not that is essentially required. And EC 2.45 is usually the industry standards, and the other one is just the two extremes. We try to push it to low end and also push it to high nutrient to look at the difference. Also interestingly, we found a low level of fertigation, so reduce the fertigation level by more than 70%, does not reduce the post-harvest quality of capsicum. So this is quite interesting. So this gives us some way of potentially grower can manipulate their fertigation to save fertilizer. Also, we found it very interesting, but still I think we need more work to be done here. High EC in capsicum actually reduce the fruit potassium and calcium content, which you all know potassium content, calcium are very important for human health. So really, this is something interesting we need to explore in the future as well. The last trial, which was more recent, cucumber fertigation, and the second half of 2022, we had two varieties growing and a lot more data, but I'll just quickly go through and give you an overview. So high and low EC do not affect photosynthesis yield, but it does change mineral nutrient in leaves. So this is similar to what we found in capsicum. And the high and low ECs affect post-harvest quality, but it depends on the genotype. One genotype, the Lebanese, doesn't really been affected much, but the continental has the post-harvest trait has been affected by the ECs. We also did some preliminary experiments on gene expression, try to look at, for example, why the potassium has dropped. So we did some experiment on looking at the gene expression, like the potassium channels, KAT3, HKT, in those cucumbers. They show a very different pattern, but you can see the expression levels are quite different. So we need to understand. That's why we will go through the other two omics, transcriptomics and metabolomics, to look at the actual underlying mechanisms for those changes. Okay, to summarize, we generally found that high electric conductivities or high fertigation does not really significantly improve yield and quality of the tested vegetable. So this is kind of slightly against the norm, because for growers usually say, okay, usually to some extent a higher fertilizer is usually better than low fertilizer. And we also found the low EC can maintain yield quality in some genotypes of the tested vegetables, not all of them. For example, one of the cucumber varieties did have some reduced quality traits. Okay, some more experiments. So we are going to start another trial in the greenhouse on cucumber with different potassium levels, as we just mentioned. EC trials are completed. We are now going to manipulating only one macronutrient. So we will do the gene expression analysis, metabolites profiling, and sensory evaluation. We hope to understand the mechanisms why those, and then provide more advice to Australian and Qatarian growers in the future. I would like to thank Future Food Systems and Qatar National Research Foundation for the financial support, and the PhD students and master's students and others involved in the project, and also the project members. And thank to all Future Food Systems CRC staff and students for the joint work and collaboration. And also, I think the students have really enjoyed their communication with other students in different parts of the CRC. Thank you.
Okay, thank you very much, Xianghua. Okay, I think we'll leave all our questions until the end. So the next speaker is Dr. Hongwei Liu. He'll talk about novel bio-based solutions for effective management of key root pathogens in protected cropping.
Thank you, David.
Okay, yep.
Need to. Yeah.
Very good.
Good afternoon, everyone. So today I'm going to talk about using Burkholderia species to control soil-borne pathogens. My name is Hongwei Liu. I'm from Western Sydney University. So firstly, I just briefly introduce about the project. This project aims to deliver innovative solutions for controlling crucial soil-borne or root-associated pathogens in horticulture crops, enhancing Australian agribusiness profitability and growth. It will develop bio-based technologies to utilize crop-optimized microbiomes, plants' immune systems, and biochemical products to revolutionize the way root disease are tackled in both protected and field conditions. We are planning to introduce unique tools to suppress disease and weed host, offering horticulture farmers additional income systems. So in this talk, I will use Fusarium pseudogrammaneum, the pathogen, as an example. It causes the corn rot disease of the wheat plant. In Australia, wheat is a major winter crop, which is has an impressive annual production estimated around 25 million tons a year. Despite of this, the agriculture sector faces considerable challenges, most notably from the fungal pathogens like Fusarium pseudogrammaneum, causing estimated annual yield losses reaching to $468 million. Particularly alarming is corn rot disease caused by the soil-borne pathogen, Fusarium pseudogrammaneum, that poses a substantial threat to our cereal industry. Despite numerous attempts, currently there's no available commercialized methods to control this pathogen due to many obstacles. It is crucial that we can focus our research efforts on understanding complex interactions between the pathogen, our soil system, crops, and also how the interactions are influenced by the climate change scenario. Bacteria, integral to the ecosystem functionality and plant performance. It has great potential for controlling soil-borne diseases. As an alternative to traditional synthetic chemicals, bacteria present an environmental friendly, effective, and economic method for pathogen control. And in my study, we isolated the bacteria from wheat plants with the aim to identify effective strains to control the pathogen. As shown in the slide, we found we isolated multiple bacteria from the wheat, and one of which could significantly inhibit the pathogen Fusarium pseudogrammaneum, and we identified the beneficial bacterium using the whole genome sequencing and the 16S RNA single sequencing. We found it was identified as a Burkholderia ambiferia. It has about 1.27 micrometers length, and displaying very promising implications for the controlling the Fusarium pseudogrammaneum. upon placing the genome of this bacterium into the context of a comprehensive bacterial database, we observe that the bacterium genome, which is about 7.4 megabase pairs, and it contains 7,361 genes, which is significantly larger than the other bacterium of the same species in the database. This suggests that our strain, Burkholderia ambiferia, has an enhanced capacity for protein production, potentially aiding its colonization on the wheat plant. This finding also aligns with existing knowledge in the bacterial genomics, which is, as we can see from the correlation graph, there's a strong positive correlation between the bacterium genome size and the number of coding genes they have, which indicates that the adaptive capacities of bacterium with larger genomes in the plant. As this depicted in this illustration, it demonstrates key mechanisms that the bacterium can benefit the plants. Among which, the bacterium can combat the pathogen via production of siderophores, and also can directly promote the plant growth via solubilize the phosphate in the soil to make it more available for the plant to use so that they can grow better. Upon further exploration of the genetic functions of this bacterium, we found the Burkholderia ambiferia has the pathways for siderophore production. Subsequent testing on agar plates also confirmed its capacity to produce siderophores. And similarly, we also found it has the capacity to solubilize the phosphate on agar plates, suggesting that it also has a potential to promote the plant growth by directly adding nutrients availability to the wheat plant. So now we identified an effective bacterium strain and also a good plant disease model or system. Then we can investigate the colonization process and how it impacts wheat plant growth. Through inoculation, the bacterium in different doses, namely 1 or 3 doses, and the sequences, which was earlier or later than the pathogen, and under varied conditions, which was interrupted or non-interrupted microbiome, soil microbiome. We have examined several key points, and we found from the 2 graphs on the left shows biomass production of the wheat shoot, and the right panel showed the biomass production of the wheat root in a microcosm experiment. We found a higher bacterial doses resulted in the increased wheat biomass production, and suggesting the higher dose application of the bacterium probably also had better soil colonization. The presence of bacterium, it doesn't matter it's introduced simultaneously with the pathogen or earlier, could all greatly enhance the plant disease tolerances. Upon further examination of the plants treated with the bacterium and the pathogen, we discovered that the expression of the plant defense genes was also impacted by both types of the treatments, which were the either by the beneficial bacterium or by the pathogens. However, these findings are based on my preliminary analysis, and the enhanced, more detailed analysis in the future are expected to provide a deeper insight into the mechanistic interactions among the plant, the bacterium, and the pathogen. Additionally, we also As shown in the red panel, we also quantify the pathogen load in the soil and the plant using qPCR analysis. Our initial results suggested a temporal pattern in the colonization of the pathogen in the plant. More definite conclusions will follow as we further analyze and validate these initial findings. And now I'm waiting for the amplicate and the metagenomics data to further analyze these processes. Overall, our findings to date highlight the potential of the beneficial bacterium Burkholderia amblyophilia in controlling the fungal pathogen Fusarium pseudograminarium in the wheat plant. That's my talk today. Thank you for your attention, and I welcome any questions you may have.
Okay, thank you very much. That was very good.
Thank you.
If you could stop sharing your screen, that'd be helpful.
Yeah, last yeah, sorry. Hey, stop share. Yes.
Okay. Yeah, that always seems to be the hardest thing for all speakers to do, is to stop sharing their screen. Very nice talk. We're going to save questions until the end. And our last speaker in program 2 is Associate Professor Kirsty Bayless from Murdoch. She'll talk about novel microbiome technologies to increase profitability for Australian horticulture. She's from Murdoch University. Kirsty.
Thanks, David. I'm
Yes.
just trying. Oh God, I've got a message telling me I have to quit Zoom to share my screen.
Ah,
I'll just tell it later.
well, good luck with that.
Okay, let's try this.
Yep. There you go.
All righty, we're cooking with gas.
Success.
Okay, lucky last speaker today in this theme. So I'm going to tell you about a project which is along the same lines, because we're all talking sort of about the microbiology of different crops. But in a nutshell, this project is about developing new products that might increase the yield and therefore profitability of Australian horticultural crops. And the approach we're taking to do this is analysing the microbiomes that are associated with these crops, all the way through from seed to harvest. We're also looking at how microbiomes might change when they're exposed to biotic or abiotic stresses. And the aim there is that we may be able to select some microorganisms from within the microbiome that are associated with that stress tolerance to then develop these into new products, and hopefully products that will assist plants that are under stress so they can continue to yield. Briefly, this is the team. I just want to point out the A team over here on the left-hand side. These are the people doing the hands-on work. We've got various different expertise from Murdoch University. I'd also really like to acknowledge our industry partners. So they're heavily involved in our field trials, Melville Park and Eco Growth. And then we have some advisory groups and other participants, and especially our growers from around Australia who are supplying samples for us. So the project's not possible without this huge team of people. Now, before I leap into what we're doing and the results we've got, I just want to talk a little bit about the focus that we have for this project. So we're not doing protected cropping. Our project is a field-based project. And the reason why is because there are actually very limited studies on microbiomes in field-grown crops. And in fact, most of them, when you look at them, are just snapshots in time or space. So research teams seem to go out into a field, walk through a crop and take some samples, randomly sampled, but they only do it once. They don't seem to follow it through the season. So much more detailed information is needed on field-grown crops. So that is the focus of this project. We are going out and we're sampling field-grown horticultural crops from different growing regions across Australia. And we want to unravel some questions like, how does the variety of the crop or the soil type or the climate or even the agronomic practices shape the microbiome? Does the microbiome actually change over time through the season of the crop? Or is there a stable core microbiome that is always there that the plant needs to grow? And most importantly, is there a correlation between the microbiome and crop performance? So the growth of the crop and the yield. Now, our other focus is that we want to look at the whole microbiome. So most studies do tend to focus just on the bacterial microbiome, and there is a reason for that. It's because there's a lot of data available that you can compare your own microbiome against. But probably only about 10% of studies have looked at the fungal microbiome, and yet fungi are so important for plant growth. Not just for killing them, there are good fungi as well. And there are also many other microorganisms that form part of the microbiome, including protists and archaea. So we want to look at the whole picture. So we're using both metabarcoding, so amplifying specific gene regions, and also metagenomics, which is whole genome sequencing. So we can get a big picture of the microbiome. And the more information we have, the more we will know about what are the microbes that are existing in the microbiomes. Are they beneficial? Are they saprophytes? Are they pathogens? And also, what are they doing? So the more information we have, the more we know about their function and how that influences plant growth. So in conjunction with Hort Innovation, who is one of our major funders, and also we have a stakeholder advisory group, which has industry, government, various growers sitting on it. We selected 3 horticultural crops. We've got tomatoes and avocados and potatoes. So 2 vegetable crops, both annuals, and a perennial fruit crop. And the stresses that we've identified, thanks to the advisory group, we've got 2 pathogens, Fusarium, which is common across Australia in tomato crops, Phytophthora, which is another huge problem in avocado plantations. And then for potatoes, we've got abiotic stresses, which in our instance, we've chosen heat and how calcium also influences heat stress or heat tolerance and the quality of the potatoes. And there's a picture of our beautiful student team there. So Henry's on the left, Sumitra in the middle, and Assad on the right. And most of what I'm presenting today, or in fact, all of what I'm presenting today, is Henry's work. So I just want to acknowledge that. So we're going to be talking about tomatoes. Now, obviously, if the main aim of this project is to develop new products, first of all, we need to know that product application actually has some sort of, reasonable result if it is applied in the field. And there aren't actually a great deal of studies that have been performed really well with lots of replication or even repetition. And strangely enough, some of them are actually being performed by the product companies themselves. So there's no independent assessment, if you like. So we're really looking at, can we start by demonstrating product application does work? And if so, is it related to the microbiome? So Henry, to his credit, in his very first year of his PhD, has done 2 field trials on 2 separate farms, testing 3 different biological-based products applied to field-grown tomatoes. He's looked at crop growth and yield, and he's also looked at metabarcoding of the rhizosphere and the soil, both bacteria and ITS. And Anish, who is our postdoc on the project, has also taken the same samples, and he's looking at the root and phyllosphere using whole genome sequencing, so metagenomics. So between the 2 of them, they've pretty much got every compartment in the crop covered, from the soil right through to the phyllosphere. We're not doing the air or the water, but maybe we'll do those down the track. So today I'm just going to tell you a little bit about the product application and what the results were from the trial. So this is shoot fresh weight, and this is the data that came from 1 farm. And I should point out, sorry, if you can hear the dog chewing on her toy in the background, I do apologise. She's the guide dog sitting there behind me. So this is on 1 farm. This farm did not apply any products at all in terms of conventional fertilizers or anything else. They just applied these biological products. So they either had 1 of 4 treatments. We had an untreated control. We had product A, which was a microbiology-based product. We had product B, which was an organic acid. And then we had product C, which was a seaweed-based product. Now, I can't tell you the names of any of those, but just trust me, that's the 3 different types of products we used. On the left, we had Roma tomatoes, and on the right, we have cherry tomatoes. So 2 different types of tomatoes as well. And this is just the fresh shoot weight. I haven't included included dry shoot weight here. But you can see there was quite an interesting result by applying these products. So in the Roma tomatoes, they responded very well with much more shoot biomass after the organic acid application, whereas the cherry tomatoes had a better response to the seaweed product. And that was actually consistent with the root fresh weight as well. So if you look at that here, again, same farm, same products, Roma on the left also again responded in root biomass with the organic acid, and cherry tomatoes on the right again had a good response to seaweed, but interestingly, the organic acid also influenced root biomass here. So what does it all mean? What we really need to know now, given that Henry's PhD is about fusarium wilt, is does applying these products and improving crop growth also confer tolerance to fusarium wilt? Now, both of the farms where we've had these trials running don't have fusarium wilt. So obviously we can't introduce it onto those farms. So for this particular part of his PhD, we're going to have to do a glasshouse trial because we can't introduce a pathogen onto a farm, as you would understand. So what we've done is we've collected the field soil from all of the plots that had product applied and the untreated plots as well. And we've brought that back to the glasshouse. He's growing tomatoes in that soil now, and he's going to infect that with fusarium. So we'll see if that soil with the amendments, which had improved growth in the plants, can actually also tolerate fusarium, or is the soil suppressive to fusarium? We don't know. Now, the million-dollar question here is what members of the microbiome are associated with this improved biomass? And if we have fusarium, we find fusarium tolerance as well, are their microbiome, is that part of it as well? So watch this space. We are literally about 1 week away from having that data available to present, but it just wasn't ready at this point in time. But all of the metagenomics has been done. But those of you who are familiar with this technology would know that data analysis from DNA sequencing is a huge bottleneck. So in about a week's time, or maybe 2 weeks' time, ask me then, and we'll be able to tell you what the microbiome is doing as well, and if there's a correlation with that growth that we saw. So to sum up, that is our project in a nutshell. Very early days yet, but it is a field-based study on horticultural crops. We're focused on metabarcoding and metagenomics to assess the whole microbiome. And we have proven that product application does actually influence tomato biomass, which is great. Now we're looking at whether the correlation of crop performance with the microbiome may be able to lead to selection of microorganisms, which could then be developed into products. So we will keep you posted. Thank you.
Okay, thank you very much. If you could stop sharing your screen, that would be very good. So that is a demonstration of at least 4 of the projects we have ongoing in our program. Let's see. What types of questions do we have here, Cordelia?
So yeah, we have a number of questions for Phil. So Phil, you're very popular.
Yeah. We have about 24 questions for
yeah.
So
what we can do here is we have about 5 minutes before the next session starts. So maybe Phil, if you want to answer one or two questions, and then afterwards the other questions Lux are going to follow up after the talk.
What I can do is I can see them here. I'll touch on them very quickly and then enter some more detailed responses.
Yeah.
Yeah.
So are our findings applicable? Findings with tomatoes applicable to other crops? If the approach works, which is still a pretty big if, then I believe that the same principle can be applied in hydroponic crops in general. We'd need to collect the specific data because there can be differences even between varieties of plants. But because of the consistency of the hydroponic environment, that's what lends itself to this kind of approach. Next one, second part first. We use mainly metabarcoding up till now, but I also do shotgun sequencing with a nanopore sequencer. So the MEDIC barcoding gives the bulk of the information, but I'm hoping to, I've done some initial metagenome analysis and hope to get some more information from that. We target bacteria and fungi, but the key microbiome for us is the bacterial one, because that's the dominant organism in the plants, and we want something that's resilient and some degree of complexity. The fungi tend to be kind of do whatever they want. How cost effective? So one of the main constraints that we've got is to make this test practical and be able to be repeated every week or two. Otherwise, it's just not useful. So we're not aiming to use DNA as the testing, probably. And this is the biggest part of the challenge. So just detecting from the DNA is relatively straightforward, but doing it in a timely, cost-effective way is a lot harder. So pathogens affect the diversity either directly or via the plant. So for instance, there are studies showing that a foliar pathogen, powdery mildew, affects the makeup of the root microbiome, and it is via the root exudates, but I don't study those directly. We just look at the net result. So which compartments did we test? This is a rock wool system. So there isn't really a air and water compartment. I take samples from the rock wool and also from the bare roots that grow outside the rock wool. And each of those has a distinct microbiome because it's a different environment. We don't look at phyllosphere at all, leaves and stuff like that.
Phil, you've answered about 4 questions, which is outstanding. I think maybe the best thing for you to do is to type your answer into that, so we can move on. I have a couple. That was an excellent talk, by the way, hence you have so many questions. I have a question for each other person, and I know we don't have time to address them, but I want you guys to think about it. For instance, for Zhanghua, we may want to think about why there's a negative effect of high EC for some of our vegetables in that crop. What is the mode of action that might be taking in place that reduces the productivity or the quality? For Hongwei, I was sort of struck by the fact that this was wheat, and wheat is not typically grown in a glass house. So you used wheat to get to your pathogen, but how might you apply this research to vegetables growing in protected cropping? And lastly, for Kirsty, I realize you're not growing your plant material inside, but rather outside. But the question that always comes to me is, if you're trying different products, what is the cost of these products? What does it cost to apply these products to the field? And what is your return on your investment? That is some sort of life cycle analysis. That may not be a major part of your research, but it will be incredibly important to industry and to growers. If it's very expensive and you get a minimal return, then it may not be worth it. So anyway, those were my questions for everyone. Yeah, that was a speed deal. Yeah, 3:50 brought us right in on time. Outstanding. Okay, well, I'll be quiet now, and I'll leave this to Dave.
Great. Dave, thanks so much.
You're welcome.
Great to be here this afternoon, and really thrilled to sort of kick off program 3. And I want to start by just giving a shout out to Kirsty and her team, because you may have picked up my name was included on the screen there, and that's via one of the other hats that I wear as co-custodian of Melville Park. And I want to underscore Kirsty's comments about Henry and his hard work. He is one of our great PhD students. So he has a scholarship from the CRC, and he's here in Australia. His family and young children, wife and young children, are home back in Nigeria. So this is a significant commitment of his life and personally to be able to be here and doing this. And I've observed him in the field and just how hard he works. So that's terrific. So to my opening remarks today, which are really just to set the scene for the next two projects. And so I'm going to pull up the screen now. Within program 3, we're looking at the value of the value-added food sector, which is about novel formulations and production protocols, innovation in specialized segments such as alternate food proteins, free-from foods, precision nutrition products, innovation in food processing, packaging, logistics, delivery, and technologies, and new science and technology for verifying product claims. And when you're talking about value to value-added foods, we're agnostic at one level, but I think we sit within trends within Australia, and there's a couple that I want to highlight. And one relates to fruit and veg consumption, and this is related to ABS data, and I'm happy afterwards to share these slides as well. The bottom line and takeaway from it, say for children, is that we completely suck at feeding our children the recommended levels of fruit and veg. We're talking single-digit percentages relative to what they should be. And particularly on the veg side, it's even lower than what the advertised numbers are because it includes processed potatoes, which are French fries and potato chips. So we're talking like 1% are getting adequate amounts. When you get to adulthood, it's not much better. We're still in the single digits, and men trail women, and we're down to under 3%. So this is, really dramatic statistics. Similarly, when we look at percentage of grains and cereals, and again, this is ABS data and very recent data, so 2021, we should be 33%, so 1/3 refined or low fiber and 2/3 whole grain or high fiber. In fact, the data shows we're completely flipped around and doing the opposite. So if you look at, we're going to add value to food, we're going to create foods that we want consumers to buy in our shops. Are we also, as a program and as a CRC, wanting to be part of the solution of dealing with our issues of why we're not eating the foods we should be eating, and what's involved in that? I've got a number of slides related to the barriers to success and why that is currently not the case now. And again, there's some really interesting data published recently from Nature Positive Production, which is in a recent book, and then also Nature Food as well. And I think they underscore why we're not achieving what we'd want to. And so there's a lot of conversation around what we should be eating, but in fact, we don't eat it. And even when we want to eat it, we don't eat it. The last piece of data I wanted to share, and this is important because, we came up with this program 5 years ago, and we're sort of in year 4 now of the CRC. And so were our assumptions right about the kinds of foods that we want to create and the technologies we want to support? So News Corp have just released not so long ago their trend forecast for 2023 under their Food Corp sub-brand, and that's built largely around taste.com. So I'm sure everyone in the audience has found a recipe on taste.com at some point. Well, News Corp own that network, and they're grabbing all of that data and mining it, and they've come up with some really, really interesting trends and analyses of it. But the couple that I wanted to pick up on were since COVID, there's been a 30% increase in entertaining friends and family at home, and it looks like this is a trend which is sticking. So we got used to eating at home during the lockdown and afterwards, and we've continued with that following COVID, and much more entertaining at home. And interestingly, the dilemma that goes with that is, well, we want to do that, but we don't in fact have enough time to cook the meals that we want to share with our friends. And so what's talked now as a trend in food is elevated convenience. The idea that quick and easy is no longer enough, but what we want is fast and effortless. And we want it still to be able to deliver meals that have a wow factor to them. And so across taste.com, one of the most popular searches at the moment are one pans and tray bakes, which is an interesting thing. The last stat I wanted to reference as we just talk about our products is a particularly West Australian phenomena, and that's looking at the processed agri-food exports across Australia and the unprocessed agri-food exports. A shout out to Tasmania here, which do the best job nationwide, albeit from a small base, of transforming what they grow into food and to value-added products. Pretty much the bottom of the heap. And that goes to one of the major projects that we're involved in program 3, which is the development of the food technology facility here in Western Australia, which seeks to address this problem. So with those introductory remarks and framing, we're going to hear from 2 terrific projects which are seeking in each of their ways, both bringing high science and great agronomy and horticulture practice to the development of foods that we want to eat that are both healthy for us, good for us, and satisfy that wow factor as well. Railing Liu, Associate Professor at Murdoch University and the part of the Australian National Phenome, is going to present some results from there initially. Over to you, Railing, and thank you, everybody.
Thank you. I'm just going to share my screen. Right, can you see that?
We can.
Okay, brilliant. Good afternoon, everyone. So as David mentioned, I work based at the Australian National Phenome Centre, ANPC, and we are quite blessed in terms of the type of analytical facilities that we have. So in today's presentation, what I'll do is to tell you a little bit about what I've done and how I utilize these advanced analytical chemistry instruments to give us better understanding of our product at a molecular level, and then how we can potentially use this information to add values to our products. And for this particular talk, the products that I refer to are these fresh produce. So the first question that I want to ask everyone is, can you name all of these fresh produce that I've shown on this slide? If you don't worry, because when I was first shown, I can only name 2 of them. So let's start by looking at the picture on the left-hand side. These are globe artichoke. And moving along to this looks like a potato, but not potato. And this is a yacon. And further to the right, something that looks like a ginger, but these are actually Jerusalem artichoke. And at the bottom, these are some, looks like overgrown garlic, but instead they are elephant garlic. And I've also put pictures of the common garlic that we commonly see, and just so you can see the size of that. So these are the produce that we will analyze in our project. And there's one particular bioactive components that is common to all these 4 different plant samples. And this particular bioactive component is, oops, I'm not sure why it's not moving. Is something called inulin, and it is actually a dietary fiber. And inulin consists of a lot of different fructose units that are joined together by a beta-2,1 glycosidic bond, and it's end with a glucose unit. And usually the way how we refer to inulin, we talk about the degree of polymerization, or DP, and this refers to how many repeating fructose unit that we have in the inulin molecules. And the DP is actually affected by the type of plant species, as well as the way how we cultivate the plants itself, including the harvesting time. So why is the fascinations about this particular compound inulin? That's because of this particular beta-2,1 glycosidic bond that makes it very unique. The reason why I say that is because this particular dietary fiber, as we consume them, we are not able to digest it. It will pass through our upper gut until it gets to our lower gut, where it's being fermented by our gut bacteria. And this is what a lot of potential health benefits can emerge out from there. For example, maybe a lot of people had heard about inulin being a prebiotic because that increased the kind of good bacteria in our gut, such as lactobacilli or the bifidobacteria. And these different type of bacteria in our gut also releases gases and different chemicals. And some of these chemicals, such as short-chain fatty acid, that also can cause a number of different potential health benefits, such as reducing the blood sugar and suppress the satiety or the appetite hormone, as well as regulate the blood lipid. In today's talk, I'm not going to talk about the mechanism behind that and also some of the potential health benefit, because there's a lot of literature available. But what we do know is that, and the degree of polymerization of inulin can impact in terms of how quickly it's being fermented and what are the type of chemical that's being released by the bacteria in our gut. So at the moment, when we analyze inulin, there's no particular gold standard methods that we can use. And currently, the AOSC method is typically used to measure the total sugar content in the product. And the number that we will give, that they tend to provide, is about the average DP. So it doesn't tell us anything about the distributions of the inulin, whether they are, what's the concentrations of each of those DP as well. Although there are newer methods that can be used to measure inulin, but some of these methods also have its limitation. For example, they have poor resolution or poor reproducibility. And one of the biggest challenge in terms of trying to understand inulin better is the ability to try to quantify each of those individual DP of inulin compound in there. This is because there's no reference material that will allow us to generate calibration curves and therefore to quantify them successfully. So what we set out to do is, can we develop a method that is quick and is also highly reproducible to characterize the inulin in the sample? And if we can characterize the different degree of polymerization in the sample, can we then quantify them? If we can quantify them, can we then also apply it to characterize and quantify all the 4 different samples that I have shown you before in the first slide? And to do this, we use one of the most expensive instruments that's actually available in the ANPC, and it is called magnetic resonance mass spec, MRMS here. I'm not going to go into detail in terms of the method development and how we can use to measure inulin, but this is just an overview of the methods and the pipeline that we use. It is actually quite simple in terms of the sample preparation. We freeze dry them and we perform hot water extraction, and then we perform purification using SPE column, and then we spit back it and concentrate the extract. And with the sample with the extract, we then mix it with a chemical matrix. And this is important because the chemical matrix can make the experiment being non-reproducible. So as part of this experiment, we compare 4 different types of matrix in order to identify the most suitable matrix. Once we have identified the suitable matrix, then we put it through for analysis, and then we can look at the data to see what sort of distributions of the inulin in the sample. So briefly, I'll show you a little bit of the results. So I've mentioned earlier on that the chemical matrix that we use is important. Here I show you 2 of the results. The one on the top, we can see that the solutions are very much homogeneous compared to another example that I've shown at the bottom, which is CHCA, that shows lots of crystallization. And these actually resulted really poor results. And for CHCA-BA matrix, we can actually see a very clear distinguish for the different type of DP in the inulin. Not only is very clear, the peak intensity is also high, and in comparison to all the 3 other matrix that we have used, the peak shape is good and the intensity is also much better compared to all of them. And because of that, we opt to use this CHCA-BA matrix. And the next step that we want to do is, can we then generate our result in a reproducible way? And also, I've mentioned earlier on that because we don't have a reference compounds to perform calibration curves, then how can we prevent that and still allow us from performing quantitative work? So the way how we go about it is to use a chicory inulin powder, and we mix it with the CHCA-BA matrix. And what we subsequently adding to do is that we increase the concentration of the chicory inulin powder and see what happened to the individual DP of inulin. Here I've shown you some of the result, and you can see that as we increase the concentration, each DP is also increased in a linear way. And we can see that the R-square value are typically very good, more than 0.9. And in addition to that, we also perform replicates of analysis, and we look at the reproducibility of each of the analyses between different samples. So we can see that the coefficients of variations is very good, which is less than 20%. And this is what's the FDA standard for developing a method. Here I've shown you the result up to DP20, but in fact, we can actually quantify the DP up to 25, and we can also detect the signal for DP that are much bigger. At the moment, based on the literature, we can see some of the methods is capable of detecting and quantifying up to DP13. So we think this particular approach is an approach that we can use to then analyze our plant sample. So here's the quick results of what we have done so far for these 4 different plants. From here on the left-hand side, which is a PCA score plots. And what you can see is that on the first component, you can see a change in terms of the concentration, and in particular, higher amount of the lower DP of inulin. And for the second component, we can and for the second component, we can see increasing in the chain length. So from this overall of the summary result, we can see that these 4 different type of plant sample actually have very different concentration in terms of the different DP and also in terms of the chain length. And here I've shown you what we call it is a RKM plot, which is just a simplified versions of looking at the DP of inulin. So each dot here represents different DP of inulin, and with interval between each dot is a fructose unit. So you will know that as we go through a different dot, that's increasing 1 unit of fructose, so it's increasing in the chain length of the inulin. And the size of the dot is also corresponds to the relative abundance of the DP. So the bigger this dot, the higher the concentration that is. And you can see that for the 4 different type of plant sample, Yacon has the shortest range, which is up to 19, relatively similar chain length for Jerusalem artichoke and globe artichoke, and whereas elephant garlic, which has much longer chain length up to 45. And we also perform quantifications of the overall inulin content, and we found that Jerusalem artichoke has the highest concentration compared to all the other 3 compounds, whereas elephant garlic tends to have a much higher concentration on the longer chain length. So in summary, what I've shown you is the method that we have developed is highly reproducible and is very fast. We can analyze each sample in duplicates in less than 2 minutes. And I've also shown you that the result for this different type of plants, we have a number of, we have a very different distributions of inulin. So what can we do with this information? As I have mentioned earlier on, depending on the way how we harvest the plant sample, we can actually affect the concentrations of the inulin in the sample. So in a way, we can use this information to then maybe modify the cultivations process so that we can emphasizing in the increased concentrations of certain DP of inulin. And at the same time, now that we know the different DP of inulin in different plant sample, it's actually allowed us to understand how it can actually have an impact on our gut health as well. And in fact, we actually just recently obtained an ethics approval for a human child to look at the different type of fiber, inulin fiber in particular, as well as compared to different type of dietary fiber and how that affects our gut health. And if you're in Perth region, feel free to scan on the QR code and to find out more about the study. And if you want to participate, we are currently recruiting for volunteers at the moment. So this is kind of the next step for our project, is to look at, now we understand the degree of polymerizations of inulin, what's the potential impact on on human health, really. So finally, I just want to thank the teams that who's been involved in doing the work, including technical supply for technical support for using the instrument, as well as hands-on data analysis. And of course, there's Future Food System and Mount Lindsay for providing funding and a different type of plant sample that I use in this particular project. So that's all for my talk for this afternoon.
Great, Railing, thank you so much. An exceptional presentation. And I just wanted to note from the photo you just shared at the end there, in the middle was Professor Jeremy Nicholson, who's the academic program lead for program 3 and who's currently traveling. But really great work. And it looks like from what it's elephant garlic's the clear winner as the data come up. And I'm sure in our question time, we'll ask a bit more about the implications of that and some of the other questions. So to Andrew Tilley now, who's going to follow on because it's a related project. And Andrew, welcome to the stage.
Thanks, David. Let me just see if I can share that.
Right, so it seems I'm the only one having trouble sharing screens today. So there you go, all good.
Just try and...
And so while Railing's presentation was dealing with very long chain inulin, Andrew's research is looking at another element within one of the same products within globe artichokes, and that's dealing with phenolic composition. Andrew, all yours.
Thanks, David. Hi, everyone. So as David said, I'm a PhD student at Murdoch University and working on the globe artichoke project along with Railing and David. So artichokes aren't a big part of Australia's normal diet. And a reason for this is because it's quite hard to prepare. So it's very labor intensive. And when you prepare it, there's a lot of wastage. So about 60% ends up going in the bin because it's not edible. But this makes it an excellent candidate for value-add processing, make a product that's easier for consumers to prepare at home and be convenient and increase their consumption. And so that's a part of what we're trying to achieve in this project. So Mark McHenry and Julia McHenry are leading this project. So their farm in Denmark, WA, they grow artichokes, and they've been selecting different artichoke lines that have a high diversity in terms of their shape, color, and composition. And so this really diverse pool of artichokes is an excellent base to select particular lines that might be better suited to that value-added processing. So you might be asking, what are we going to be selecting for? Well, artichokes are very good at going brown very quickly. So in a value-added product, if an artichoke goes brown, that's not only visually unappealing for a consumer, but it also decreases the nutritional quality as the phenolics are degraded into brown pigments. Now, we already have a number of different mitigation processes in the industry to deal with enzymatic browning. We can apply heat treatment, pH regulation, or modified atmosphere to inhibit the enzyme polyphenol oxidase from forming those brown pigments. But this, to an extent, is potentially limiting what sort of value-add products we can develop to an extent. Another option is we can select particular cultivars that have a low propensity for browning. So that is usually because they have a low phenolic content and a low enzyme activity. But obviously we're trying to achieve something that's more nutritious. And so reducing the phenolic content isn't something that we really want to be doing. So one of our questions was, can we achieve a product, have a particular line that is low propensity for browning, but has that high phenolic content still. And so that's what I'm going to talk about today, some of our findings that are helping us achieve that. So just quickly, an overview of what enzymatic browning is typically described as. So phenolic compounds that have this diphenyl group here, oh, don't do that, diphenyl group are oxidized by the enzyme polyphenol oxidase, and then they undergo subsequent polymerization reactions until they eventually form a brown pigment. This is the way that most enzymatic browning processes are described in literature, but we actually know very little about this polymerization step here, how it's occurring, and what the main contributors are to it. And this is a problem because it's a critical part of that brown pigment formation. So moving on to what we've done and some of our findings. So in 2022, we harvested artichokes and we cut them into quarters, and we then measured the change in color over 90 minutes as they went brown. And to do this, we used a colorimeter, which is essentially a handheld spectrophotometer that presents color as 3D coordinates in a 3D space. And so using this, we can track the change in color on that cut surface over time by measuring the change in the position of those coordinates, either individually or as a whole. We then extracted the phenolics from the artichokes and we quantified the two main ones that are always present in artichoke using liquid chromatography mass spectrometry. And those two phenolics are chlorogenic acid, which you might recognize from coffee, and a dicaphyl-quinic isomer, which is structurally related to chlorogenic acid. Now, the important thing about these two compounds is that they both have that diphenyl group, which suggests that they can be substrates for polyphenol oxidase to act on. So we then looked for some correlations between these variables to see if there was any relationships between them. And the biggest one that stood out was the correlation between the ratio of chlorogenic acid to dicaphyl-quinic acid and those browning variables. So in the top right graph, you can see we had a positive correlation between the ratio and the change in lightness after 90 minutes of browning. Now, essentially what this is saying is that if you have a low ratio, then you see a darker pigment in your browning. In the bottom graph, it's a negative correlation between the ratio and the total change in color after 90 minutes. Essentially, this is saying that if you have a low ratio, then you see a larger change in color during browning. Now, we didn't really understand whether this was a legitimate relationship, whether it was something going on, so we investigated. And to start with, we took pure mixtures of chlorogenic acid and di-CQA, and we mixed them at different ratios to represent what's in the artichoke. We then added in polyphenol oxidase that we extracted from artichoke, and we followed the change in color in a spectrophotometer. So on this graph, you can see a couple of different lines. The purple line is just di-CQA by itself with the enzyme. The red line is just chlorogenic acid by itself. The green line is a 1-to-1 ratio of the two together. That blue line is a 2-to-1 ratio. And then that orange line you see in the middle is just the sum of the red and purple lines together, so a hypothetical value. Now, the spectrophotometer is measuring the absorbance of light, but the color that we actually see with our eyes is essentially everything that isn't absorbed. So you can see all of our mixtures were absorbing roughly between 400 and 540 nanometers. So this is your purples, blues, and greens. Just close that. But the colors that we're seeing are the complementary ones on the wheels. So that is your yellows, oranges, and reds. Now, the interesting thing here that we've noticed is that the orange line is what we'd expect the absorbance to be if we mix them together and there's no interaction. But because we can see our blue and green lines are above that, it's indicating that there's a synergistic interaction between those two compounds that's leading to a more intense brown pigment. So we wanted to see what was happening to these two compounds during that incubation period. And so to do this, we took the same mixtures at a slightly diluted concentration, and we tracked the concentration change of those compounds over 60 minutes. So in this first graph, you're looking at the two compounds when they're just by themselves. So that red line is just chlorogenic acid, and we can see a nice typical decrease in concentration over time as it's oxidized by polyphenol oxidase. But we can see the blue line, which is dicaprylquinic acid isomer, is showing very little change over time with the enzyme when it's by itself. So this suggests that it's either not a substrate for the enzyme or it's a very poor one. When we mix those two compounds together at a 2:1 ratio, we see a very different picture. So the blue line, which is the di-CQA, is now decreasing very rapidly over time. And we can see that the chlorogenic acid is still decreasing, but at a reduced rate. And this trend continues at the 1:1 ratio. So again, very rapid decrease for di-CQA and an even slower decrease for chlorogenic acid. So di-CQA cannot be oxidized by PPO unless another substrate is present. And the rate of decrease for chlorogenic acid decreases proportional to the concentration of di-CQA. So there's obviously some sort of interaction between these two compounds that helps produce a more intense brown pigment. And we want to see whether they're both contributing equally to that brown pigment formation. So to look at this, we extracted phenolics from artichokes, we quantified those two phenolics, and then we diluted them down to the same concentration as that first assay. We then spiked some of the extracts with chlorogenic acid to achieve different ratios within the same extract. We then repeated the experiment where we added the enzyme and measured the change in absorbance. So you can see on this graph, the red line is a 100 micromole concentration solution of the two at a 1:1 ratio. The green line is the same ratio, but at twice the concentration. You can then see the purple dotted line in the middle. This is just the hypothetical halfway point between those two lines. And the blue line is a 2:1 ratio. So we've spiked the lower concentration one with more chlorogenic acid. Now, if both of those compounds contributed equally to the intensity of the brown pigments, we would expect the blue and the purple lines to be the same. But we can see the blue line is slightly lower, and this is suggesting that the di-CQA is actually contributing more to the intensity of the brown pigment than chlorogenic acid does. So what does this mean for the artichokes? So di-CQA looks like it's not a substrate for PPO, or it's a very poor one, but it is still contributing substantially to that brown pigment formation. And this doesn't really fit that model that we talked about earlier, where it was very much a linear one. So we, in a recent review paper that's under review, we proposed a new mechanism for browning, where a PPO substrate, such as chlorogenic acid, can oxidatively couple to a non-PPO substrate, such as di-CQA. This can then be reoxidized as part of a redox cycle by another chlorogenic acid molecule, and this would allow for subsequent polymerization and formation of those brown pigments. So what does it mean from an application point of view? If browning is obviously more complicated than we normally anticipate it to be, then there's potential for more exploration and identifying new ways of mitigating the process. We already have some mitigation processes in industry, but they are to an extent potentially limiting what sort of new products we can develop. For artichokes in particular, if we select artichoke lines that have a high ratio, this may help minimize the extent of browning that we see during the processing of value-add products. Thank you. I'll stop sharing now.
Thank you so much, Andrew, for that great presentation. Just looking at questions, none have come up, so I get to ask two. Railing, first to you. You mentioned that the equipment you were using was some of the most expensive equipment in the AMPC. So how does that work, do you think, for the future commercialization of these assays? And is there a way to say, could you imagine in the future a cheap and easy way to detect degree of polymerization? And then why would we want to do that as well? I need to unmute there. Sorry.
Sorry. Yes, we chosen the most expensive one because that's giving us the best resolution. But now that we have developed the method and we think there are other equivalent type of mass spec method can also be used to perform the same analysis. And that's partly why the way how we perform the extraction and all the whole process are relatively simple. So that will enable translations of that methods using a different type of mass spec instrument. So this is not going to be specific to the kind of instrument that we have used here. And in terms of why do we want to do that is because we know the different chain lengths of inulin can potentially have a different health benefit because it's how quickly it gets to our lower gut and how then is being fermented by the bacteria, what type of bacteria is in our gut, and subsequently what type of chemicals that is being released. And those are important. And more importantly is all these different type of plant produce is inulin is a dietary fiber. Your introduction earlier on saying that in Australia, we are really under-consumed dietary fiber, and not just in Australia, but it's actually globally in a lot of different countries also have the same issue, not just in adult, but in children as well. So understanding these potential very good or healthy dietary fiber that could also help us to introduce another type of dietary fibers for people to choose from. And because we now know the detailed molecular information of these type of fiber, we can also provide a more specific guideline or information to the public health to say, if you are eating dietary fiber, but not any dietary fiber, there are specific type of dietary fibers, and that could potentially giving you what type of health benefit. So that's kind of more longer term type of goal for us. Now we have first step understanding at a molecular level, the food that we eat. Now the next step is for us, how does that going to have an impact on Earth? On us as an individual, and then how we can then translate that. And in fact, as part of this human study, we also work with the chef to see how we can then produce the food and make it in a simple meal that individual can use it. And as part of the study, we also say that how much we want to give it to people and see we can start to see a response. So that's quite a number of different angles where we can contribute to give us a better understanding of the whole product itself, really.
Yeah, Raylene, thanks so much. And it's such a terrific example of the use of what is very sophisticated high science all the way down to then how that can make a difference in how people just choose a meal and what they eat. So thanks for that. Andrew, in yours, you again, through analytic means, you're looking at different levels in artichokes. Did you in your data see variation in different plants? You mentioned at the start that Mark McHenry was growing different shapes, colors and sizes. So he's got a fair bit of phenotypic expression across his artichokes. Were you seeing differences within those artichokes in terms of their browning?
Yeah, definitely. So we saw differences over the season, but in terms of the actual phenolic content, we saw very big differences over the season, but also just between individual plants. So it could range from 8 milligrams per gram on the dry weight for chlorogenic acid all the way up to 30 milligrams per gram. So there's a lot of diversity that can allow for us to pick plants that have a high phenolic content. We can then look at the composition and select ones that have a likely low propensity for browning as well.
Yeah, so again, what I can see here is the use of really sophisticated analytical chemistry mechanisms that can then inform back to breeding at an agronomy level and saying, well, these artichokes look the same, but in fact they're not. Or this one's a bit more purple, that one's a bit more green, or this has got spikes on the end and that one doesn't. But you're able to delve into the chemistry of that and say, yeah, but this one may be better for you, or from an appeal point of view, do better in post-processing. So when you cut it, it doesn't brown as much. Is that now? What do you think about the chances of being able to then give that information back? And do you think that Mark would be interested in then selecting for those that are showing a more promising result?
Yeah, I think it's obviously a long game when you're talking about plant selection and breeding. It takes a number of years, but if you're at a point where you're going to select plants, you can then identify the ones that are best suited for whatever factors you want, and then breed them for a larger crop down the line.
Right. Okay. Thank you both so much for the presentation. It's really excellent. Cordelia.
So that concludes the research showcase for today. Again, thank you so much. And if you do have additional questions, please feel free to reach out to the CRC. We are more than happy to connect you to the relevant speakers. If there are topics that you are interested in, please reach out. The session is recorded and the recording will be made available to you. So thank you very much. And also, just before we go, I just want to acknowledge the CRC team who has been doing all the organizing. They have done such a great job, and thank you especially for Luke, who's in behind that Future Food System logo there. Thank you.